I'll send you a video going over that. In fact, if you subscribe to my YouTube channel, you could go through the videos and find the video that describes this. The answers are already posted online. Most of the stuff I do, the content, the whole year's content, most of it's already online, right, on the YouTube channel. So there's really no reason, but I can see if I can find it and then send it to you. If not, I'll make one today during the day sometime and post it. It'll go through the answers that those classic experiments in biology are very important, okay? Because they let us know, and you've started doing your homework, I hope, your chapter homework, because that, I kid you not, you have no time left. You have to keep up with your homework, do as much of it as you can, as early as you can, get your reading done. There's not going to be a lot of time. There's no makeup, there's no resubmissions, right? No late work, because all the work this week is due on Friday. Next week, I'm not accepting anything late. Whatever I don't have by Friday is it. So if you want to retake something, you better schedule it with me during an advisory, because after school every day this week, we're busy, right? And we got a field trip on Thursday. I got Think Box every other day. I mean, it's a busy week. Yeah?
All right. Is one wet? No.
This about to be a long.
Say yes or no.
Oh, no. There's nothing wrong about it. I'm not answering that question. It's nothing to do with what we're talking about. So, yeah.
It's about the thing you gave us yesterday.
If it's a quick question, I'll try to address it.
So when they injected the mouse with the heat killed smooth and rough. I forgot. Wasn't that?
Could I have somebody's work? I'll do a short run through it. I'll send you the. Just give me somebody's work. I'll look through it. I remember the assignment. Yeah.
If this strain has died since it can be detected.
Right. So let's talk about that real quick. It's going to be a quick version and the long version, which is going to go through the details. All of these experiments will be on the final. All these experiments will be on the final. So know this you have it. I'm not even going to give you a grade for it. It's yours. You're going to get a quiz on it tomorrow. I'm going to go over it today. Everything in this packet will be on the final. I'll pull some questions right out of these experiments. Are we clear? Each of these experiments is going to have at least one question on the final. All right. So there was a time where we knew these things, DNA, right? And by the way, in the chapter, it clearly goes over what the structure of DNA is. Why would I not go over it here? Because we've already done it. We spent a whole month on biomolecules, and you were tested on it, and you were told you were going to see them again. And now in this chapter in the book, they go over it again. And you have some homework on identifying the difference between DNA and RNA. And that's going to be a real big deal this week and next week. Your final, I will have a study guide for you this week. Traditionally, you have a week. A study guide is due to you a week before the final. You have study guides already. You filled them in. For all the chapters in the book that we've covered, you have a notebook that's full of study guides that I've given you all quarter long. There really is no excuse for, but I'm going to give you a very clear, what I like to call a test blueprint, which is going to break down what this final is going to be about, how many questions, and what form each question is going to be, and what topic is being covered by each question. It's called the test blueprint. So I'm going to go beyond that, beyond the syllabus, then the study guide, and I'm going to give you a test blueprint. All right? But I'm going to wait on that because I need to get it done, and I got a lot going on this week. I have a lot of your work I have to grade. It's over there. That's your chapter 8 work, all right? And some chapter 7, some late work from chapter 7. I'm going to get that graded this week too. Also, some of you need to retake the test. You have to schedule that retake with me this week during advisory. Am I clear? I'm posting this so that we are all clear and that there's no question that I said it. So you should be writing it down. Now, so they go over what the differences are between DNA and RNA and proteins, but we're going to talk about it here as well. But around the time of 1920s and 30s, we knew there was this thing called RNA. We knew there was this stuff called DNA, and we knew there was this stuff called proteins. We knew what they were made of. We knew that RNA had 4 nucleotides. We knew DNA had 4 nucleotides, and we knew that they were almost identical, except what? That's right. This one had the U, and this one had the T. Those were the kind of the big differences. They both had A, they both had C, and they both had G, right? So they both had A, they both had C, and they both had G. But DNA had the T, and RNA had the U. They knew that. They did. The biochemists figured that out. They identified them. They labeled them. They named them. They knew that there was in a test tube. They could isolate it. They knew it had this test tube. And when they looked at it in this test and when they did their chemical tests, they identified 4 different things and they labeled each one of those 4 different things. That was pretty good work. Took a long time to figure that out. They didn't know what DNA and RNA did, though. They really didn't understand what they did. They knew what proteins were. They knew there were 20 amino acids in living systems. They knew that. And they knew there were there were all kinds of different qualities, right? Some were polar, some were non-polar, some were charged, some were big, some were small, some had ring structures, some were straight, some were bent. All kinds of different structures, all kinds of different groups. So there was a lot of, right? Which one do you think had the most amount of information? Proteins had a lot of information, right? If you're just looking at information, you're like, this one had the most information in them, where these had kind of standard, right? These two were kind of the same, right? Maybe a little different because one had a U, the other a T. But you only had 4. If you were going to build a book of life, a book that defined every part, or at least structurally defined every part of you, right? Something that was like, I need to send this. This is the book that I'm going to send to my children and my children's children from now until the end of time. This information, I'm going to send that information somewhere. In which one of these 3 would you use as your alphabet? The one with 4 letters, the other one with 4 letters except it has a T, or the one with 20 letters? The protein. I would use the protein. And most scientists at the time thought that proteins was the inheritable material. Proteins were where the book of life was written, right? That all your genes were in proteins. That's what they thought, and it made sense, right? It made a lot of sense. But it turned out it was DNA. DNA was the stuff that held all the inheritance. The genes were in the DNA. Those things that Mendel talked about that we'll cover when we get back from our winter break. That stuff that Mendel talked about, those little inheritable things that the peas sent that made the peas tall or yellow or whatever that you covered in middle school. All that stuff, it turns out, was DNA. Passed on from one person to the next, from one parent to the next parent, from one flower, one plant to the next plant in the lineage. And it turns out that proteins were not the stuff of life. But how did they figure that out? How did we figure out that it wasn't the proteins, that it was the DNA? How did that happen? And that's really an important experiment, isn't it? Isn't it important that you know how we know that DNA is the stuff of life, the stuff that sends information from one generation to the next? So what they did in the experiment, right? And who remembers what they did in the experiment?
They stuck the mouses with they injected the mouses with that
Bacteria, okay. What did they do with the bacteria then?
they injected it in the
They put it in dishes.
Put it in dishes and put the mouse on them. Some die, some stay alive.
That's right, that's right.
I didn't get that one, an unexpected one. I didn't get that one because it was, like, when they added both of the bacteria together, rough strain,
the thing died.
Right.
smooth strain.
Okay, yeah, it's confusing. I hear you. It's confusing. It's not an easy. It's actually an elegant experiment. It's a beautiful experiment. The results are clear, and you get real quick results. Sad, a mouse dies, right? But it's very clear what the results are. So you have this bacteria. It's a pneumococcus. What do you think pneumococcus do? They produce what? Pneumonia. They produce pneumonia. And so that's one way you can get pneumonia. Now there's a viral pneumonia, but it's a bacteria. So bacteria, is a cell, right? So we know it's a cell. We know, you should know that by now. It's going to be on the final. So you see how I'm saying these things? If I were you, if you're a student in college and a professor says that, you're writing that down. So you have a bacteria cell. You know it has kind of a little DNA thing. There's no organelles that have membranes inside. There's protein. There's little ribosomes. There's a little thing. There's little plasmids. That kind of thing, right? So it's a bacteria, but it's not this shape. It's not oval. In fact, the word coccus means it's circular. So it looks more like this little circle, little things like that, right? And there's two kinds. There's this smooth kind, and then there's this rough kind. All right, so I'm gonna just put little red dots on it. And it turns out that the smooth kind was the deadly kind. Do you remember that? The smooth kind was the deadly kind. They did experiments. They say, okay, if I put the rough kind into the mouse, then the smooth kind kills it. If I put the rough kind in the mouse, then the mouse doesn't die. And the reason for that is these things here, and just so you know, and I explained it in the paper, okay? These that you read, hopefully you read it. These little rough dots on the outside, this is a cell, right? That's a cell membrane. What are the rough dots? What makes it rough? What are the dots? No, they're on the membrane. Ribosomes aren't found in the membrane, the plasma membrane. What's found in the plasma membrane? Good guess. Kind of enzymes of proteins. They're integral membrane proteins, right? That's what you find in a membrane. Phospholipids and proteins. You should know that by now, right? Phospholipids and proteins are in the membrane of the cell. If you have these proteins, these red proteins, well, the mouse's immune system can identify them. So when you have these proteins, it turns out that these often proteins that are on the outside of the cell are how you identify who you are. When we go over genetics, and that's going to be one of your winter break packets, okay? He's going to be going over some of the stuff. Yeah, you have like 3. Yeah, you have like. It's 3 weeks. You have 3 weeks off, 1 a week, or you can do all 3 of them quick. We can spend our class time doing this if you'd like, talking about this rather than going over the content. But that's up to you. About how many packets we have over winter break. But we have. You're not even listening to yourselves, though, to me. But in any case, you know that there are 3 blood groups, right? There's A, it's actually 4, B, O, and then there's AB. And we'll talk more about this, but I need you to understand that the reason, if you have A blood type, the reason you have A is because on the surface of your proteins you have these kind of, I'm making them into triangles, but you have these little green triangle proteins on the outside. And so when your cell sees these cells with green triangles, what does your body do?
Nothing.
Nothing, because they say, hey, this is me. I'm okay. This is part of me. I'm good. I'm cool. It's like in the old days when I was a kid, you had the Bloods and the Crips, and they had their colors.
We still got.
I mean, not like they were when I was a kid. When I was a kid, they were all out. Everybody was everywhere. But today, but your cells are the same way. They go and they identify, are you part of me or are you not part of me? If you're not part of me, they attack. They kill it. So if I take a pencil and I stab you with it and break the tip off in your body's going to go to it. Your cells, your white blood cells are going to go and try to sense it. Once they sense it's not part of you, they attack it. They kill it. They try to kill it. It's dead already, but they try to kill it, and they'll push it. Literally, it happened to my brother when he got a splinter. Over time, your area will get real red, and it'll literally push it right out of your body, almost like in a movie. Of course, it takes weeks to do it, but it's called, you might even get an infection, but the infection is when these cells multiply faster and your body can kill them, right? So if you're a B blood type person, then you have these, you have these kind of, let's call them circles on the outside. So the A is triangles, B is circles. O blood type, you don't have any proteins on it. I'm an O blood type, and we don't have any proteins. We don't have any of these particular proteins, right? And because of that, we're called the universal donor. So we could give our blood to anyone because we don't have those proteins on the outside of the cell. So no cell is going to read it and say, oh, this isn't me. They're going to read it and say, well, I can't believe you're on your phone, dear. I cannot believe you're on your phone. Your notebook is not open.
It is OQT, George, really about to learn.
Okay, you need to leave. Go outside my room. Take your stuff. Go outside the room. Go to the office. Do what you got to do. Don't come back with a jean jacket on. And when you come back when you're ready to learn with a notebook open and not your phone in your hand. Thank you. Yes.
Is there any blood type that is
Say that again.
is there like a blood type? Like, is
Well,
there-
let me get to the end. Let me get to the end before you ask another question. So B blood type has these, I'm going to call them circles. They're not circles, right? And they're not triangles, and they're not green, and they're not blue, but they're proteins. They're on the outside. O blood type doesn't have these proteins on the outside. So when a white blood cell, if I give you my blood, your body looks at my blood cells and doesn't see any proteins on the outside, so they're cool with it. There's no identifier. So again, I like the gang analogy, because it's like if you're wearing red and you go into a Crips neighborhood, you're going to get in trouble, right? Or if you're wearing blue and you're going into a Bloods neighborhood, you're going to get in trouble, right? Because they're going to identify you as the opposite, as not being part of that particular group. Well, your red and your blood cells are the same way. So anytime you give an A gives blood to an A, everything's cool because they're in the same group. They have the same identifiers. B is the same thing. Those are cell surface proteins, right? B has the same. It can give to B, but it can't give to A because A will say, "Whoa, no," right? O doesn't have any identifiers, and so O can give to anybody. But you know what? O cannot take from A or B, right? Because if you put A blood type in me, my body's going to say, this is not me, attack. If you put B blood type in me, my body says, that's not me, attack. So I can only take blood from people with O blood type.
So,
Wait, 1 second. Now, people with AB blood type, and this is why we discussed this topic in genetics, they express two different genes. Obviously, there's a gene for A, there's a gene for B, there's a gene for AB, okay? People with AB blood type, they have both triangles and circles. So they have a parent who has A blood type and a parent who has B blood type, and these genes are both expressed. So this is not a dominant recessive situation. This is what we call codominance, and we'll talk about that after winter break. And in codominant situations, you see both genes showing up. So both sets of proteins are expressed. So people with AB blood type can take blood from A. They're called universal acceptors because they could take blood from A. Why? Because they have the A identifier. So their bodies can't tell that A is not part of them. The B, they could take blood from B because they have the B identifier. They could take blood from O because O doesn't have any identifiers. So they're called the universal acceptor. They can't give to anybody except AB.
So can you use another
I'm not sure how to use that identifier except that it would be like if you walked into a Cribs neighborhood and you were wearing a blue hat, then you kept walking, you're going into a blood neighborhood, so you'd switch hats to a red hat. So, or if you rather think of it, you have a hat that's one half's red, one half's blue, right? So other than, and then if you're an O, you don't have any hat at all. Does that make sense?
Yeah. Is there such thing as O negative?
Yeah, we'll talk. That's another story, but it has to do with a smaller protein that's also an identifier. But so the people that have A blood type have this genotype. They have a double A genotype. People that have B blood type, they have a B genotype, and people with O have what are called they have both genes are broken. They don't have A or B. All right. Where AB obviously has AB genotype. Does that make sense to everybody? So that's how cells work. Well, it turns out that this cell here, these two cells, the smooth strain has no cell surface protein. So if and the rough strain has these red ones. Well, when you take and inject this red one to a mouse, what does that mouse do? It kills it. The mouse says, hey, wait a minute, that's not me. It's dead. I'm going to have to kill it. And the mouse's immune system kills that bacteria. But you take this smooth strain and you inject it into the mouse, it dies because there's no cell surface protein to tell it what's going on. How did it get those proteins, though? That was the question. This is, by the way, how we know that DNA is the inheritable material for life. So the way that they did it in the experiment, again, very beautiful experiment. I need you also to know that control groups are going to be heavily tested on the final. You guys did horribly on your essays overall, identifying what was the control group or what was the experimental group. In fact, if you did less than 3, if you scored it, there were 5 possible points. If you scored less than 3, I didn't even put the grade in your grade book. I left it out. So if you scored a 3, 4, or 5, then you got your points in the grade book for the essay. So it's not that I didn't grade them, it's that you guys obviously have demonstrated you still don't understand what a control group is. Some of you are having trouble reading a graph. You're still having trouble reading a graph. It's a real problem. So if you don't see a grade in that essay part of that test, you need to see me after some time to really have a deep discussion over how to read a graph and what is a control group and what's an experimental group. We're not going to do it here, though. All right, so you have the smooth and the rough. Those were the 2. So what the experimenters asked was, what's being inherited? How come they had an idea why the smooth and rough, what the difference was in their lethality. And so, of course, what they did is they took the rough strain, they injected it, the mouse lived. They injected the smooth strain, the mouse died. They killed the bacteria with heat. They killed the bacteria. They injected that into the mouse and it lived. Why would they do that? Why would they kill the bacteria by heating it and then inject the dead material into the mouse?
To see if they would have some type of reaction. To see.
We call that a control group. A control group is something we're using to compare our results to. So we have these mice, and there's 3 of them, right? In fact, let me do something else rather than trying to draw them. you have these mice, this is a control group. So you have these mice, and I know they're horribly drawn, deal with it. And you have 3 of them, or actually 4. To 1, we're doing an experiment, right? We want to see what's being inherited. We take and we put the rough strain RS in there, the mouse lives. There's 3 people talking. I'm not going to mention names, but when I have a parent conference and they ask me why you're not doing well, I'm going to say, let me refer you to this video. Let me tell you that the person I'm talking to in this video was your daughter. And you need to understand that if she's talking in class, she can't understand what's going on. Reasonable conversation, yes?
Yeah.
So rough strain goes in there, the mouse lives. What does that tell you? That they're not deadly, right? And then I inject the smooth strain. Even if you don't know about cell surface proteins, you know the smooth strain dies, right? It kills the rat, the mouse, whatever. Then you know that then there's something about that smooth strain that makes it deadly, right? But then I take and I do a control, because what were these? What were the rough and smooth? They were also controls, right? Because I knew the smooth killed, and I knew the rough didn't. But why did I inject mice? Why did I even inject a mouse to kill the mouse if I knew it was going to kill it? I need to have something to compare it to. So these 3 are controls. This is what I call my positive control. This is called the negative control. Why is this positive control? Because if I'm saying the smooth bacteria have this deadly trait, then I have to prove that they're deadly, and I have to show that this kills mice. And this is it. This is what I'm going to use to compare it to. And then if I think that the rough smooth doesn't, I have to show that. That's a control group. So now I heat kill them. I kill the bacteria, and I inject those. And guess what happens? It lives. And what does that tell you? That means the bacteria have to be alive. So so then I take the heat kill. I'm going to go HK if you don't mind. And I take the smooth strain. I'm going to go at SS, smooth strain. Oh, no, sorry. Smooth strain heat killed, right? So I should say that. These are both smooth strain. I take the rough strain and plus the heat killed, I inject them, and guess what happens? I let them sit in it dies. What does that tell you? What did that experiment just say? By the way, this is your experimental group. This is the one you did not know what was going on. You couldn't predict it. You knew the smooth was going to kill them. You just did it to use a comparison group to make sure that your smooth strain were actually deadly and your rough strain were not. You wanted to verify that. So that's why you did these 2 control groups, the positive and the negative. And you heat killed one. It was another control group that you had to make sure that you could show heat killed by itself doesn't kill anything. Does that make sense? But now I mix the heat killed with the rough strain, I inject it, now it dies. What do you think happened? What's your hypothesis?
That the bacteria died.
Say again loudly.
That the bacteria died.
If the bacteria died, what's killing the mouse? It's not a toxin. How do I know it's not a toxin? Because my control group, my heat killed control group, right? These are killed. I inject everything in that test tube into the mouse, and guess what? So I know there's nothing poisonous in there. That's why I have this control group, to show that it's not something, I'm not injecting these things with some kind of poison. That's why I do this control group, because I got to show that there's no poison in the sample. So now I take these heat killed, right, and I mix them with rough strain, and then all of a sudden it kills them. Kills the mouse.
So I guess.
Not too much bacteria, but good guess. The same amount was put in all the test tubes. Yeah. But it's a good guess. That's one of those factors that you would control, right? You keep the same, because you're only changing one thing, that you're mixing the heat killed with the rough strain. Yeah.
So when the heat killed, because in the third one it said the heat killed when it was injected into the mice lived because it was already dead. So if they injected the dead one and the mice was able to live, the rough one wouldn't the immune system be able to detect that one, so it's still...
Right, so that's right. See, oh my God, that's exactly the kind of thing you gotta be doing. It's perfect thinking. You have your rough strain is going in there. So according to our experiments, the mouse's immune system should be able to identify this and kill it. We know the heat killed is not killing the mouse. Why? Because we have this one that says it lived. But something's happening, right, that makes this combination deadly. What do you think is happening?
It's they don't.
They changed. What changed? Right, the live strain must have changed. So what does that mean? That means that somehow this bacteria that's rough, right? This rough bacteria somehow got some kind of inheritable material from the dead one, right? There's these pieces, they're dead, right? There's just all these pieces. Some of this material here must have gotten into this cell and changed it, right? So it changed it. It's the only thing that they could think of. So somehow this bacteria changed from rough to what?
Smooth.
To smooth strain. Somehow. Some genetic material changed the rough to smooth. So what was that genetic material was the question. So then came the second part of the experiment, right? So here's what they did. They said, okay, well, what if we have these enzymes. Enzymes, remember, they speed up reactions. Well, RNase, what does RNase do?
It breaks down the DNA.
Excellent, 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 excellent. I love that you're thinking like that. It means you did your homework, you did your classwork, but you got a little bit wrong. It's not DNA, it's what? Look at it carefully. RNA. It breaks down the RNA. So it takes RNA and it breaks it down into nucleotides. So no more RNA. No more. So you get rid of the RNA. It's breaking it down. Protease does what? It breaks down proteins.
Yeah. DNA is breaking down DNA.
That's right. No more proteins. And DNA, do you see how beautiful this is? Because now you have some tools that let you get rid of DNA, or get rid of protein, or get rid of RNA. So they took this, so what they did, then these are your tools. These are tools. These aren't really experiments, they're tools. So now you take your heat killed, right? Heat killed, rough, smooth strain, right? SS, smooth strain. We know that there's inheritable material. We know that they can pass down that smooth trait to their friends, right? So they take and they put it into 3 test tubes, right? And you should be able to get a lot from this, including the answers that you're going to need for your final. You're going to add RNase. You're going to add to this group protease. And then you're going to add DNase to this group. Oh my goodness, what does that mean? Then you're going to take and you're going to mix this one with what? The rough strain, right? Mix this one, whatever's left over, to the rough strain. And then this one to the with the rough strain. And then you're going to inject it into mice.
Oh, and then you see which one.
Right?
Which one? Oh.
Then you're going to inject it into mice. Yes, I know they're horrible drawings. I don't want to hear it. Say what now? I can't hear you.
The mouse died.
Could just made a circle.
It could be a roller coaster, but it is a mouse, so like I said, deal it.
Are you showing that the mouse died for the first two?
Yep, that's right, we'll look at it, but you'll see what happens in just a second. So in the first with the first mouse where you got rid of the RNA, hold on a second. You got rid of the RNA in the first one, right? And what happens? The mouse dies. Could it be that RNA was what passed on the genes for smooth strain to the rough strain? Could that have happened?
Yes.
Could it be that RNA is the stuff that's being sent from the heat-killed bacteria to the rough strain to make them smooth strain? Could it be that?
Yeah.
Yeah, gotta be. So
Yeah.
you saying that RNA is making rough smooth?
No, I'm asking you, is it?
I think it got
Possibly.
It could be it could possibly.
Look at this and answer. Think first. Think about it. RNase does what? So I add RNase to the heat killed. I then mix them with the rough strain, and then I inject and the mouse dies. I ask the question, is it possible that RNA is what sent the information to change the heat killed? Or the rough strain? No, why not? There's no more RNA. Excellent. There's no more RNA. It can't be. Look, I love you guys thinking. Look at it. There's no more RNA. No more. Why? Because we added RNase. So this experiment shows it can't be RNA, right? So then I add protease to the sample. And I inject the protease. The protease means it got rid of what? Proteins. And the mouse dies. So it can't be protein. So the inheritable material can't be protein. Man, I thought for sure it was going to be protein. That's what scientists probably thought back then. I can imagine sitting down, reading your journal or the paper at their coffee. These biologists spend their whole life studying this, reading their journal at their morning coffee, their newspaper. They're like, oh, what? It's not protein. I can't believe it. So yeah, it wasn't protein, and that was a shocker. Because remember, it had the most, that's the alphabet that all of us said we would use proteins for the alphabet for the book of life because there's so many letters. But no, that's not it. Hold on to your questions. Then they added DNase. And they had to make sure there wasn't something else that was sending, maybe it wasn't any of these. Maybe it was something else that was sending the information to the rough strain to change them. So they added a rough strain, and what happened? They lived. The mice were happy. The scientists were ecstatic. Because what did they just identify?
That was the...
DNA is the inheritable material.
So DNA is what makes the mouse alive?
The DNA is what but remember, here's the tricky part to this experiment. It's a beautiful experiment, but think about it. I didn't say you added DNA. I said you added DNase. DNase gets rid of the DNA.
Right.
And I got rid of the DNA from the heat kill, from the smooth strain, and now the rough strain stayed rough. They did not inherit the material. But only because I got rid of the DNA, proving beyond a reasonable doubt that DNA was the inheritable material. Now, did I really prove it? No, but I had collected enough evidence to make people confident that DNA was the inheritable material. As it stands, we're pretty sure, after all these years and experiments that we've done, remember this is back in the 20s and 30s and 40s, etc., we're pretty sure that this was definitely DNA is the inheritable material. Well, it's 9:54. I promise I wouldn't try to take things all the way to the last minute. I think I talked more about this experiment than I really wanted to. But I think tomorrow's discussion needs to happen. because tomorrow the Meselson stall experiment is very important in DNA replication. I can tell you that both of these, I've seen that both of these in various standardized exams. All right? So I think it's worth our time to have that discussion tomorrow. How do you feel about that? Do you agree? Do you feel more confident now about this experiment? So go back and make sure you understand it. Could you talk to each other about it? I promise you this experiment will be on the test. I promise you there'll be about 2 or 3 questions on the final about this experiment. On this test coming up, on the next test, which will be Monday, not Friday, on that test, it's going to be a big part of that test, but you'll see it again on the final. So work on it during advisory. Come and ask me questions. Catch up. Make sure your work is getting inputted this week into the Jupyter grades so that no one's grades. I want next week to be just us worrying about the final and whatever's left of DNA and RNA, and DNA replication, or transcription, translation, and DNA replication. Are we good? All right. All right. So what did we do? What did we do? So we took, I know you're misunderstanding this based on the questions you're asking me. Oops. I know you're misunderstanding because the questions that you're asking show that you didn't understand 2 things. Number 1, you confused how the bacteria kill the mice. Now we know that's corrected, right? It's not an immune reaction. It's that the mouse could not identify the bacteria because of the smooth texture of their outer layer. Their outer membrane was not readable by the mouse's immune system because they had some kind of slime layer that protected them. Is that understood? That was a result of some information in that bacteria that allowed it to produce that layer. That information, every time it went through cell division, that information was passed on to its progeny. Every time you made a cell division, 1 became 2, 2 became 4, 4 became 16, right? Every time you did that cell division, that information was passed along. Just like your parents passed information to you, and they got it from their parents and their parents before them, et cetera, et cetera. Okay? Where the other problem is that people didn't understand is what is heat killed? What is heat killed? So heat killed means, what they meant by heat killed is that they took the smooth bacteria, they had it in a test tube, they heated it up so to the point where the cells broke apart. There were no cells, there's nothing alive in this test tube now. They were alive, and now they're not. After you heat it up, they're not alive. And in this mix, you had 5 things. You had DNA, RNA, proteins, lipids, and carbohydrates, right? All the parts that make up the cell. You don't have any cells anymore, though. They're all broken apart. That's what we meant by heat killed smooth. That's what it meant. Break it down into its component parts, the 5 parts. So they're sitting there in the test tube. Is that clear? Are there any questions about that? Okay, so now you have the heat-killed bacteria. Now, you took that test tube, a sample of this test tube, and you poured it into this test tube that had what? Rough strain, the rough strain of the bacteria. That meant it looked like this, right? Now remember, every time you have a rough strain, when it produced progeny using binary fission, right, splitting 1 into 2, it was rough. What are its progeny? Rough. What are going to be their progeny? Rough. It's because that's the information they're inheriting. So all these rough strains, every time they divide, they're rough strains. They don't change unless there's something else happens, and we're going to talk about that in just a minute. So these 5 come in here. These 5 parts were all in a mess. There's no cells. They're all in a solution, right? You took some of that. You added it to the rough strain, and what happened to those rough strain? They turned to smooth strain. So some of the information that was in here that made this smooth must have gotten into these cells, right? some of that inheritable information got into these cells somehow. Made them into smooth state, made them deadly, right? The only question they had is which one of these 5, where was that information? Was that information in the DNA, RNA, protein, lipids, and carbohydrates? Remember, they thought it was in the protein.
It's really in the DNA.
But we know today it was in the DNA, but how do we know that? That's what's going to be the question on the test. How do you know that? Do you understand how the experiment worked? So which one was the inheritable information in? Which one of those 5? They thought it was either DNA, protein, or RNA, and they were pretty sure it was in protein. We know today it's wrong, it's in DNA, blah, blah, but it's not. Right? Because it had the most number of options, and if you're going to make a book of life, odds are you're going to use the language that had the most number of letters, right? I mean, that makes sense, because you have all these details to iron out. English language has what? I can't remember. Is it 26 letters?
Yeah.
26 letters. Well, protein had 20. DNA only has 4. RNA only has 4. All right. So when we look at heat-killed smooth strain, we're talking about a test tube that has just those 5 components in there, DNA, RNA. That's it, right? No cells. Then we take those and we add RNase, which is doing what? Is breaking up RNA. So after I add this, is there any RNA in that test tube? No RNA. So that means in this test tube, there is protein, there is lipids, there's DNA, right? There's carbohydrates, but there's no RNA. Does that make sense? So now I take that RNA. Remember, what are we asking? We're asking, where is that information that makes the rough cells into smooth cells? Where is that information? Which one of the 5? We got rid of one of those 5, right? We only have 4 in here now. We add it to the rough strain, let it sit for a while. If the information is in one of those 4, right? If it's in carbohydrates, lipids, DNA, or protein, what is going to happen to the rough strain? It's going to what? I can't hear you. Say it louder, I can't hear you. Decode it. Yeah, it'll decode it. If the information is in, it'll be decoded. It'll coat the cell. It'll make it smooth. That's right. Good. Yes. And then the mouse dies. Yes. So the information's in there because the mouse died. So which is it? It's not what? Because there was no RNA in there. We got rid of it. Does that make sense? Then another test tube. We have another test tube over here. And in this test tube, we got rid of the protein. So the protein's gone. But we have the DNA, the carbohydrates, the lipids, the carbohydrates, right? And we also have RNA, don't we? We have some RNA in there too. But there's no protein now, right? Why is there no protein in this one? Because we got rid of it. Proteases, that's what proteases do. So now you got those 4 going in there. Well, again, if the information's in there somewhere, then it's going to coat the bacteria and the rat's going to die. Well, the rat died. The mouse died. It's not a rat, it's a mouse. The mouse died, right? So, could it be a protein?
No.
So it can't be RNA, because this one showed us that. It can't be a protein. This one showed us that. But it can still be DNA. But why do we, if we think it's DNA, why do we even bother doing this? Yeah. So you're for sure, what else could it be?
It could be something else.
What other things? Carbohydrates or lipids? It was highly unlikely, but hey, we thought it was protein, and we just showed it wasn't, so we better make sure. So we run the DNAs. So that got rid of it again. We still had a test tube, and we still had we had the RNA, we had the lipid, now this time we had the protein this time, and the only one we didn't have was the DNA. So now the DNA was missing. We still had 4, but it wasn't DNA. And we added that to the rough strain. Let it sit for a while. And then if the DNA had the information, would the rough strain have the information to change into smooth? No, because we got rid of it. So we got rid of the DNA. So therefore it doesn't have the information to turn into smooth. So what would happen to the mouse? It would live, and it did. So that's why we were pretty sure back then that DNA was the information of life.
So the DNA did not turn smooth, right?
It did not turn smooth, so that when you injected the rough into the mouse cells did what? Killed the bacteria. They recognized.
But the other two on there,
They did change.
Okay.
And what's the only one that these two had that this one didn't? DNA. That's how we knew DNA was with all the information of inheritable, the information you can inherit was in DNA. That's how we knew it. Does that make sense? An amazing experiment, beautiful experiment, one of the most beautiful, elegant experiments ever. There's a few of these magic experiments that are just like simple and gorgeous and just clear. It's exciting. So yeah, we're going to go over the next experiment. It's Messel's install, and I think it's Messel's install. As I said, if you better be doing that chapter reading, and not wait till the last minute and get those assignments done. There's no makeup on these assignments.
These assignments that you want next week.
Yes, and the faster you do them, the more in depth you're able to go, the more questions you can ask, the better your grade. I mean, so Messelson install, they looked at. Okay, so now we know that DNA is the inheritable material. How does it make copies? We know there's only 4 letters. And we know there's billions and billions of letters, though. We know there's billions and... I mean, that number is not something that we can understand. I mean, I've been thinking about it. I probably have a better picture of it than you do. I've been thinking about it for many, many years. But I can't imagine that you have a clear picture of what billions and billions and billions of nucleotides, 3 billion nucleotides, 3 billion letters. That's a lot of letters. That's a lot of information. How do we make accurate copies of that? Because every time there's a mistake, what do we call that? If I have A, T, G, C, T, by the way, is this DNA or RNA? DNA, how do you know? As a T, there's no U, that's right. G, C, and we go on for a billion nucleotides. What happens if you make a copy of this and it's A, C, C, G, T, G, C, etc.? We just changed something. What do we call that change, people? You know it. I wouldn't ask you if you didn't know it. What do you call that change? You know this. You've grown up with it. It's a word that's used everywhere.
What's the first letter?
When you're changing the letters in DNA, what do you call it? When you make a mistake, when your replication system makes a mistake, what do we call that? It could be an accident. We do it on purpose in the lab, but yeah, it's certainly that it's not meant to happen.
Mutation?
It's called a mutation. That's what a mutation is. It's a change in the DNA. We're going to talk about several kinds of mutations, but mutations are just a mistake in the copying of the message. Imagine I gave you a dictionary and I said, a dictionary would be very small compared to this, all right? And I told you, copy this dictionary. I said, copy this dictionary. All right, so I tell you to copy the dictionary. How long would it take you to copy the dictionary? Hours,
Unless
days,
you talk about
weeks,
some writing,
months,
you talk about
handwriting.
hours.
I'll let you type it. How long do you think it would like to take to type out? To type, I would say
A week.
personally,
A month, a week.
I don't know. I can't even guess. We can calculate it. We could calculate it if you could do like 20 words per minute. If you work for 8 hours a day and you took an hour break and you have a dictionary as whatever. We could figure it out. But let's not even worry about it. Let's just say it's a long time, right? If you had a time limit, if you were going to do it, would you make mistakes? Yes. I think there would definitely be mistakes. That's why you have mistakes in your books, right? Because somebody somewhere is going to get tired and they're going to repeat something. I make mistakes on these videos, right? You make mistakes in your homework, you make mistakes on the test. You're going to make mistakes when you're copying it, right? Mistakes are going to happen. No, very few individuals can do anything perfectly all the time. In fact, I'll say no one does everything perfectly all the time, right? Okay, so mistakes are going to happen. So mutations are always going to happen, right? Because now here's the question. It would take us months, and some people say years. So, okay, to copy a dictionary. Billions of nucleotides is longer than the dictionary, right? It would probably be something like a couple hundred dictionaries. I don't know. I'm just guessing, making guesses here. So let me ask you this. How long do you think it takes for your cells to make copies of this DNA? Okay.
I think it's like
We measure it in this. Minutes. It takes 20 minutes for a bacterial cell to make a copy of itself. And to make a copy of its DNA, it probably takes less than 20 minutes, probably 5 minutes or 10 minutes. Because it's 20 minutes for the whole cell to completely replicate. We measure how long it takes for your DNA to make copies of itself in minutes. If you cut yourself, it'd probably take a week for it to heal. That's billions of cells millions of cells making copies of themselves, not just their DNA, but the cells themselves, and filling in the gaps, right? Restructuring, rebuilding itself. I don't know what you're saying. I can't hear you. We have what?
dead skin.
Why do we have dead skin? That's another story. But are we clear on this idea that cells are able to do these copies? And do you think they're going to make mistakes? Absolutely, they're going to make mistakes. We call those mistakes mutations. Now, do they happen often? Oddly, amazingly, like it's a miracle, but it doesn't happen as often as you think. And not only does it not happen often, but the cell can actually correct its mistakes as an autocorrect. It corrects its own mistakes. That's crazy. It makes sense, right? If you're going to have all these people, otherwise, every time you had a baby, you don't know if you're going to get a puppy or a tree or a fish. How do you know what you're going to have unless you're sure you're not going to have too many? Unless you're sure you're not going to make too many what? Mistakes. Because if you're going to have changes, right? If you're going to have mutations, it's going to change what you have, right? And the baby you have. So if there's changes in your genes, if those mutations happen, that means there's going to be changes in the next person born.
So that's how like kids might become Down syndrome.
That, yes, Down syndrome is a good example of a mutation that occurs. It's actually a chromosomal abnormality, but it's not really a change in the DNA sequence. It's a change in the chromosome, how many chromosomes you have. Down syndrome is trisomy 21, so you have one extra chromosome. So it's a little bit different, right? But yes, it's those mistakes happen, and then the result is that the child. But that's also why some children are born with tails. Some people are born with 6 fingers. Some people's earlobes are attached. Why are we different? That's also why some people are dark skinned, some people have light skin. We all started as one, let's assume you're religious and you started with Adam and Eve, right? If we all started with Adam and Eve and everybody looks like their parents, right? Right? If everybody always looked like their parents and we start, why are we different? Why are there differences? Why are there people that with Asian features and African features and European features and Aborigine features and Native American features, right? How did we get those differences? Mutation. So whether you're religious or not religious, I don't really care one way or the other. My point is, that no matter how you look at it, you started with a small group or two people, right? And then that slowly expanded to this diversity that we have on the planet today. That diversity comes from this source. That source is mutation. So we over time have changed. Our DNA's changed. We're slightly different. The changes are extremely small. Less than 1%. Chimpanzees have 98.99% the same DNA as you and I. Right? And so our differences from chimpanzee are small. Can you imagine how close we are? We're virtually, just very, very small differences between you and I. Now those small differences come from mutation, that's true. But they're small because our replication mechanism is very accurate.
So what's our difference with a banana?
Our similarity, I think it's like 50% of our DNA we share with a banana. Well, why, and then remember we talked about why that was, right? We said, well, how is it that we can have any DNA that's the same with a banana? Because we all what? We all what? I don't want to say your name, but if I have to take that cell phone away, you're going to be very upset. I've been asking you 3 times to please put it away.
So you said bananas are our ancestors?
Huh? No, bananas are not our ancestor, but there was a cell back in the day, we've talked about this forever, right? The first cell, one of the first cells, before there were chloroplasts, before there were mitochondria, that DNA is still in us. And bananas, because they split. One of its children developed a chloroplast and a mitochondria. Another group of its children only got mitochondria. Those mitochondria ones became us, animals, right? The plants became a whole other group. But we share that common ancestor billions of years ago, right? We share that common ancestor, so that explains why the DNA can be found in a banana and a human, right? And see, that's the beauty of this explanation of these events that we call evolution, right? Because that's what a theory is. A theory is an explanation of facts. And this explanation called evolution is pretty solid, has a lot of evidence behind it. And one of those pieces of evidence is this fact that we all share this DNA, these little DNA sequences that are millions and billions of nucleotides long with such a diverse group of organisms. All right, so anyways, these mutations, they happen at a predictable rate. We actually know we know many regions of the DNA. We know how quickly they mutate. Some mutate very quickly, others take a long time to mutate. When I say very quickly, I mean many generations. We don't have mutations every generation happening all the time in any one place in your DNA. But you have mutations happening all the time. Because even though they're rare, we're still talking about even if they happen once in a million, we have 3 billion nucleotides. So they're gonna happen, right? Does that make sense? Yeah. That explains why there's always a winner in the lottery, right? The lottery, what are the odds that you're going to win a lottery?
1 out.
It's ridiculous. It's more likely you're going to get hit by a meteor. Yeah, it's more likely you're going to get hit by lightning. Much more likely you're going to get hit by lightning than you're going to win the lottery. That's crazy. But somebody always wins. Why? Because a lot of people play. And they play more than one. They play because it might be 1 in a billion that you win. But there's billions and billions of tickets bought, right? Or whatever. I don't know the numbers with the lottery, but you know what I'm saying. Yeah?
Did this mutation occur more likely if let's say you and another person share the same gene?
No, it doesn't happen more likely. It's not more likely. You mean that you and your spouse share the same genes?
Yeah, let's say like
No, you'll see, let's not get, it's not a bad question, it's just, let's wait till we look at DNA replication. All right, so now let's quickly, I have to get to this experiment, so it's 9:38 and we're kind of running out of time. So sorry, I want to get to your question, but I want to get through this first. So there were two ways that they could there were actually three ways that they thought DNA could make copies of itself. The first was a semi-conservative. So we knew there were two strands. We talked about that in class, haven't we? So we know there's two strands, a double helix, right? Hydrogen bonded to one another, two molecules, hydrogen bonded to one another. We know that the matching is A to T, right? And we know it's G to C, right? And we know there's 3 hydrogen bonds between G and C and 2 with A and T. So we know that that's going on, right? We know they're nucleotides. We know there's a sugar phosphate backbone, and that's this, right? The sugar phosphate backbone. We know it's 5' to 3' on this side, 5' and 3' on this side, where it's going to be 3' is their opposites. We know they're anti-parallel. All right, so we know all that. So now the question, how do we make copies of this DNA? Like, how do we take this DNA and then make a copy of this DNA? Well, they thought there was 3 possible ways. The first way is that we'd have semi-conservative. I'm going to change the color on this. This is where your colored pencils would come in handy. I'm going to go ahead and just change the color because it makes it easier to see. So let's call one strand red, one strand yellow, or blue. So in the semi-conservative model, when you make a new copy, what are you going to need to make? First of all, can I ask you this question? If you're going to make a copy of this, how many different strands are you going to need? This is 1 chromosome, right? You need 2 strands, right? So there's 3 kind of ways you can do that. The semi-conservative is you're going to have 1 new strand, right? 1 new strand, and one old strand and 1 new strand, right? Does that make sense? And then it's semi-conservative, so that means that the other strand is I don't know why I didn't switch. So that means that this other strand is also going to be copied, but then you're going to have 1 old strand and 1 new strand. So you see you need 2 new strands each, right? The yellow's the new, and the red and blue were the originals. Does that make sense? So that's one way that could happen. I see some conversations going on. I really would love it if you just would focus on what I'm talking about. I have seen many questions on the end of course exam come from this information. Another way of making this kind of a copy of these 2 strands, making 2 new molecules with 2 strands each, is called the conservative model. So in the conservative model, you would keep your original strands, and then what would happen is you'd make a new one, right? Both the 2 new strands would go together, and the 2 old strands would go together. That's called the conservative model. So this was the semi-conservative. And so why do they say semi? Because only half the old molecules with the new, you have 1 old molecule with 1 new molecule. See that? Where here you have the 2 old molecules together and the 2 new ones together, so we call that conservative. In other words, you're conserving the original. Does that make sense? You have to know this. This is going to be tested on the test on Monday, and it's definitely going to be on the final, and I've seen it on the end of course exam. So I've not seen it personally, but I've seen it on the practice exams. All right, so semi-conservative, going to be important to you. So there's either semi-conservative, conservative, and then what's the other option? Can anybody think of the other option? How else can we make a copy of it? You get what? Well, the word they use is dispersive.
Half new, half old.
The half new, half old, that's called semi-conservative.
No, it's like,
Half new, half old is the first one, right? The second one is conservative, where you got the 2 old ones stay together, 2 new ones stay together. So what's another option? It's the only other option that I can think of. Mixing them together, that's right. So you get some old strand and then you get some new strand. Oh, okay, I didn't understand what you were saying. So some old and then some new, right? So you can mix them together. If that's what you meant, then you were right. You got, it's hard to, and then of course the other strand, you get some old and some new, right? Does that make sense?
Yeah.
So that's kind of the three ways they could think of that you could get these new molecules. You can copy these things. They knew they got copied pretty well. They knew that they got copied, that the copies were, they knew that the copies were accurate, but they just weren't sure what, they weren't sure about how they were copied. Were they conservative, semi-conservative? And this was important because understanding how they got copied allowed us to do things like cloning, right? Because today we've cloned a sheep. Everybody knows that, right?
Yes.
We have companies that are cloning dogs. You love your dog, you want to keep your dog forever? They'll clone your dog. It won't be your dog, right? But it'll have the same genes. They have all kinds of things they're doing. We clone bulls. If you have a great prize bull that has all the characteristics you want in a bull, they're really meaty and they produce a lot of meat because you can have big babies. Big babies means more money. They don't eat as much, right? Right?
They're gonna be cloning us
Well, there's a whole debate about that. But let's talk. Yeah, I hope not too. But we have dispersive, we have conservative, and we have semi-conservative. So those were the three questions. Which one is accurate? So they had to design an experiment that would let them figure that out. And here comes radiation again. When I was younger and oops. All right. The next experiment, the last experiment that you have in that, and you saw how important these experiments are to your test. Tomorrow's work is going to be just as important. Meselson-Stahl. Their question was, okay, we have DNA, and we know that there's two strands. We know it's a double helix, right? Actually, I'm not even sure. I can't remember. Well, let's just go ahead and just say, we today know it's a double helix, right? We today know A and T, 2 double hydrogen bonds, right? We know C and G, 3 hydrogen bonds. We remember that from August to September, right? So this is and if you don't remember that, chapter 12, section 1 talks about DNA. Get to doing your homework. This is the third day you've had that homework assignment. If you're waiting till the last day, you've got problems. So here's this DNA double helix. And this is a chromosome. This is what we call a chromosome. When you see chromosomes, it's this, with some histones that are wrapped around, but that's another story. So you have these 2 double helixes, 5' to 3' on the other, on one strand, 3' to 5' on the other strand, right? If you're going to make copies of this, how many strands are you going to make in the copy? 2 strands each, right? There's 2 sets, 2 strands each. Do you agree? Their question was, how is this replication happening? By the way, think about this for a second. The human genome is about 3 billion nucleotides long, with a B. It's 3 billion letters. A, T, G, C, T, T, A, G. Incredible number. More than an Oxford unabridged dictionary. More letters that are in a giant dictionary. Now, what if I gave you a giant dictionary, put it up on top, so giant that it's 50 pounds, and I put it on your table, and I said, copy it. There's a room full of paper, here's a pencil, start copying. All right, 2 questions to you. Assuming you're going to do it, and I bet you I can get I bet you I can get the person who said they wouldn't do it to do it. I bet you I could. I would tell her mother that it's important that she does it for her grade. Just don't speak. I'm trying not to let anybody know it's you. But I would figure out a way if I wanted you to do it. There'd be some way. I don't know how. Maybe I'd give you an incentive. Okay, so anyways, we're getting off track. So I tell you to copy it. How long do you think it would take you to copy that dictionary? Not quite 5 million years, but it would be a long time, right? A couple years, maybe, I don't know, but it'd be an extremely long time. And that's less than 3 billion letters long. How long do you think it takes your cells to make copies? Make a guess.
9 seconds.
Half a second.
How? To make one copy. No. 12 years.
Somebody says a couple hours.
5 years.
2 hours.
6 months.
Yeah.
5 years, because like when you like 5.
Well, it takes E. coli, which is a type of bacteria, about 40 minutes to copy its bacteria. About 40 minutes to copy 40 million letters.
I wish I could.
About 1 million letters a minute.
Okay, so one letter was hit.
So 1 million letters a minute. Eukaryotes with a human chromosome has about 150 million base pairs. Right, 150 million base pairs.
Wait, what's this, like,
150 million. I said a billion for 3 billion. The base pairs are AT, GC, right? Yeah.
For the thingy right there, that's what, how is it at the top, you don't have the A to T, or is that supposed to be there?
Say that again.
At the top, for A, you have AT, GC, T, GC. Is there supposed to be an A? Isn't there supposed to be an A in between the C and the T? Or is that supposed to be like that?
Oh, it's exactly right for a reason. I'll get to it. So, a single chromosome has about 150 million base pairs. And to copy the human DNA, when your cells replicate, when you make one cell, it turns into 2 cells, it literally takes them about an hour. It takes you about an hour to copy the DNA,
anything?
your cells. It takes about 40 minutes for the bacterial cell to copy. It takes you an hour to make 3 copies of 3 billion nucleotides. 3 billion letters. In an hour. Boom! You are some lazy people. You can't copy a dictionary in faster than two years. And it does, and your cell can do 3 billion nucleotides in an hour. Well, of course you're right. I mean, it's just joking when I say you're lazy. Yeah, well, that's good that you recognize that, but that's not really the point. The point is that this is really, really an amazing event. Making copies of DNA is an amazing, incredible process that happens incredibly quickly. I hear conversations. I move people around for a reason. I will be moving people around again. Honestly, what I'm saying has a point. There's this sequence of DNA. A, T, C, G, C, T. Well, if you're going to make a copy of it, it has to be the same, doesn't it?
Yeah.
So if you were going to copy that dictionary, and by the way, that's how we used to make copies of books, right? Books were written by hand. And like the Bible, like the Quran, right? Back in the day, it was you etched the information in stones. Wait, girl. Or you wrote it in mud tablets and then you cooked them in an oven to make them stones so they wouldn't change, right? So it took a long time to make these copies. Obviously, this is not writing a letter, so it's much faster. But let me ask you something. If you were going to copy a dictionary, do you think you would make mistakes?
Yes.
You certainly would make mistakes. It would happen.
Jump over all the words.
You better have somebody editing it, huh? And checking your work and saying, oh, that's a mistake. Throw that away and start over.
Oh, no, not start over. Erase
Yeah, no, there was no erasing back in the day.
It was ink.
It was all ink. Yeah, I can't imagine. I would not want that job. I agree with all of you. That's too much. Exactly. And the thing is, do you think that when your cells are making 3 billion copies, or a copy of 3 billion letters, do you think they're going to make a mistake?
No.
They do make mistakes all the time. It happens all the time. And the amazing thing is that your body corrects the cell corrects the mistakes quickly, almost as quickly as they make them. And most of the mistakes are corrected. But what's really interesting, well, let me ask you this question. Let me ask you another question. What do we call those mistakes in copying? We call them mutations. So instead of having A, T, G, C, T, G, C, the cell might make a mistake and say A, C, C, G, T, G, C. That change is called a mutation. That's why we're all different. Did you ever ask yourself why we're all different? That's one of the main reasons we're different. Mutations. We all started as this, let's assume that you're religious. We don't have to be religious. Let's just use this as a thought experiment. Let's assume that you believe in Adam and Eve. There was one male, one female. Why don't we all look like Adam and Eve? Mutations. We should all look the same, shouldn't we?
No.
If we all start, and by the way, if there weren't mutations, we should all be the same. Period. I don't care if you're religious or you're not religious. It doesn't matter. If you're not religious, you have to think about it. We all came from an original organism. That original group of organisms had a certain set of genes. We should all have the same genes as them if we did not have mutations. We should all. Why are some of us light-skinned, others not? Why are some of us have blue eyes, others brown eyes? Why are some of us tall, some of us short? Why are some of us fat, some of us thin? Why are some of us boys and others girls? Because we have mutations. Mutations change our DNA over time. Yeah. What do you mean we can predict it? How does a cell correct mistakes, if that's your question you're asking? Is it reads the original and it says, wait a minute, this is not right, and it goes back and changes it. It doesn't really predict any future mistakes. And can we predict certain regions of the DNA are going to be more volatile? Only because we can see that there's many changes in a given amount of time. Yeah?
But that don't, like, you said that the top one is the original, right?
Uh-huh.
That's not the original.
Why is that
You
not
only
there?
have two A's, you only have one A in there. A goes with T, G goes with C.
Oh, I see what you're saying. No. I was looking at one strand here. That's a good point. Yeah, her point is that if you were looking at it, it should be this, right? Because they should match. There's two strands, right? And then this one over here should have, right?
Oh, I get it.
But what I'm saying is that if we're copying the strand, we have to have it open, right? We have to look at this, and then we make a copy of this. This is what it would be.
Oh, okay.
If there was a mutation, this part's wrong, isn't it?
Oh, okay.
So we're copying it. In order to copy it, you have to open this up, don't you? Because you can't read it if it's not open. In order to copy a book, you have to open it in order to read it so you can copy it. All right, I'm going to have to ask some of you to leave. I'm sorry, I can't. I'm running out of time. I've been having a lot of patience. It's 3:06. I really want to get through this experiment, and everybody wants to leave on time. All right. So, yes, you're right. This is not matching here, so that wouldn't work. But this is what happens when you copy it. Because when you copy it wrong, what happens to the next strand over here that's going to be made to make it be a complement, it's going to be wrong too, right? All right, so
Wait, so they have to match, or
they what now?
what she's talking about, like they have to match and stuff?
They have to match, right? If they don't match, then you don't have hydrogen bonding. If they don't have hydrogen bonding, they don't stay together. So can there be a mistake? Do they have to match? Over time, yeah, they're going to have to match. Are we good with this? Can I go on?
Yes.
All right. Yeah?
Why does
in a way, it does it differently, but let's not talk about that yet, okay? It's what you're talking about is transcription. We're talking about DNA replication. How do we copy DNA? One thing at a time. Then we'll move on to transcription, and then we'll go on to translation. Read your chapter reading, please, and thank you. I don't know what else to say to you. If you're not going to read, you're not going to understand. Because I only have 15 minutes a day, and we don't have all quarter to talk about one topic. You have to do your homework, ladies and gentlemen. This better be a really good question, sir. Go ahead. No. We're in the middle of something. Sit down and please, unless it's an emergency, and you really better have it really better be an emergency. So, here you have this DNA molecule, double-stranded. We have to make copies. There's 3 ways they thought you can make copies, 3 possible ways to make copies of this DNA molecule. The first way is called semi-conservative. In other words, you open it up, right? If this is the original, the blue with the red is original, right? You open it up, and with the blue, you make a new strand, and the new strands are in yellow here, okay? You add a new strand yellow. The new strand's yellow. The original blue strand's still there. Does that make sense?
Yeah.
So in the end, you have one blue, one old strand, and one new strand. We call that semi-conservative. Of course, the other strand was red, so you have one red strand, which is the original, and the other's yellow. That's the new one. Is everybody good with that? Would you do me a favor and close that door? Yeah. So, one old and one new. We call that semi-conservative. There's 2 strands. Half is old, half is new. We call that what? Semi. Semi means half. Conservative means you kept the original. So semi-conservative is half the DNA is old, half the strands are old, the other half new. Are we good with that?
Yep.
The other option is that they thought of was conservative. So you open up these strands, but you make a copy of each strand, the 2 new ones go together, and the 2 old ones go together. We call this conservative. In other words, the originals stay together, the new ones stay together. That's another way it could happen. It's possible. I see people staring off into space. The third way is called dispersive. Dispersive. And in dispersive, maybe instead of one full strand being old, both full strands being old, both full strands being new, or one old strand being combined with one new strand. Instead of that, maybe each new strand was partly old and partly new. We call that dispersive. So those were the 3 models that they thought could have happened. They weren't sure. They weren't sure which one they had. So they had to do an experiment. Let's research. We used a lot of radiation to tag, to identify. Remember where that whole oxygen, radioactive oxygen on the other tests? Well, we used to use a lot of that radiation. Today we use a lot more fluorescence, but we still use radiation as tags because they're easily detectable. And it's an element, so you never get rid of the element, right? Except when it breaks down naturally. So the one thing that we knew DNA had, right, if you remember what DNA is made of, the one thing we knew is called what is DNA nucleotide called? You learned the name of it. Why would I label it NB? Was it? It's a base, nitrogenous base. That's right, nitrogenous base. So, very good. I'm glad you remember. So it's a nitrogenous base, which means it has what? What element? Nitrogen. It has with nitrogen. Proteins have nitrogen as well, but for sure DNA and RNA have nitrogen. Well, we're worried about DNA, so what they decided is to use 2 different forms of nitrogen. They used a nitrogen 15, one isotope of nitrogen, which was heavier, and then you have the nitrogen 14, which is not radioactive. So nitrogen 14, which was lighter. So nitrogen 14 is lighter, nitrogen 15 is heavier. So remember that the idea of a centrifuge that we talked about? You know, when a centrifuge is like a washing machine spin cycle, right? And it spins round and round and round and round. And as the test tube spins, if this is the test tube pointing out from center, as a test tube spins around the circle, right, all the stuff goes down to the bottom, right? So if it's heavy, it goes down to the very bottom. If it's a little lighter, it goes down, but it doesn't go down as far as the heaviest. And if it's a little lighter, it goes down, but it doesn't go down as far as the middle one, right? So you can get separation in a test tube by what? By mass. The more massive the particle, in this case, the more heavy the nitrogen, the further down in the test tube it would be. Does that make sense? So this is a test tube, this is a centrifuge test tube. That's the basis of this experiment. So they had two control groups, right? And so they grew, the first thing they did, now we're talking about bacteria. So bacteria, what we can do with bacteria, and we're going to be talking a lot about this in two different classes. We have these things called Petri dishes. You've heard of these?
Yes.
And there's this stuff called agar. And agar is like Jello gelatin, but it's thicker, it's more stiff.
Like Jello?
It's like Jello, you know? You've eaten Jello before, haven't you? Like strawberry Jello. Yeah. Okay, you might think it's nasty, but you've eaten it. I don't care if you like it or not. I love it. But you like it or not, you know, it's irrelevant. The point is, you know what it is. It's like it's a semi-solid. You can be liquid, and then when it cools, it's a solid or semi-solid, and it kind of vibrates, right? Well, if what we do we use agar, which is a seaweed base, has a seaweed base, gelatin, right? It's not Jello, it's gelatin. And the gelatin forms, we pour it into the Petri dish, and a gelatin forms a growth area. What we do into the Petri dish, we can add whatever we want to the liquid before it hardens. We can do whatever we want to the Petri dish before it hardens. And so what we can do is we can put food into this gelatin. The food that we can put in are things like blood. Why would we put blood in there? So it's a good food source. We could put but we could also put things like antibiotics. We could put anything we want. So it allows us to do a lot of experiments. Yeah.
Can we make this announcement real quick?
No, I'm literally in the middle of an explanation. I'm sorry. Can you come back later?
Oh my goodness.
Like literally.
Miss Perez, so.
I tell her in a little bit, or you can leave it here and I can make the announcement before they leave, however you want. Thank you. So Petri dishes these are called growth plates. So they're growing. We use them to grow what? What are we growing? Bacteria. We use them to grow bacteria on there. All right. So if we can grow bacteria on this, that's really great. So if what we put in there, if whatever the food source is, whatever the food is, if that food has nitrogen, only nitrogen 15, then guess what's going to happen? Then the bacteria are going to have what kind of DNA? Nitrogen 15 DNA. If I have another plate that I put nitrogen 14 into the plate, right? What kind of DNA are they going to have? Nitrogen 14. So that makes sense, right? So when I did it and I run my control groups, after I take those bacteria off, they grow in these little colonies called plaques. By the way, that's why they call, when you go to the dentist, they call what grows on them plaques. Because they're bacteria, little bacteria growths. So these bacteria, if it's nitrogen 15, right, they're going to grow. And what we can do is collect them. We can break the cells apart, similar to what we did with the heat killed, right? And all we'll get is all the different components in a test tube. We're not going to have the cells, right? And we can run them in a test tube. We can put them in a test tube, and the nitrogen 15 shows up here. The nitrogen 14 shows up there, the DNA. Is that making sense?
Yes.
So we'll see a band up here if the DNA has nitrogen 14 in it, and then nitrogen 15. If it has nitrogen 15 DNA, then it's going to be down lower. Okay. So now we understand the process, right? No questions that I can see. You might have them a little later. That's okay. Raise your hand. So now let's go ahead and talk about what this experiment looked like. The first thing is you have to understand you have these Petri dishes. They're little plastic dishes they had in the back. We've used them in class before for different reasons. And the Petri dish is like a plate, looks like a plate. In fact, when we put bacteria on it, we call it plating. And in the Petri dish, we pour this liquid that turns into a semi-solid. It's a gelatin. It's like, you know, you've eaten gelatin before, seen people eat it. It's called agar. And we put nutrient in it. So we call it nutrient agar when there's nutrient in it, right? So this is called agar. It's liquid when it goes in, and then it turns into kind of a semi-solid, like a gel. Right? Everybody understand what a gel is? Yeah. Understands what a gel is? It's made of seaweed. Agar is made of seaweed proteins. They cross over and they bond together and they form kind of a... But in the agar, you can have all kinds of nutrients. And so you can put a food in there. You can put a food in this. It could be food, it could be blood. You put blood in there, the bacteria eat the blood. You put all kinds of food that you can control what you put in there. Does that make sense?
Yeah.
So you can grow these bacteria in two different ways. You can grow them using nitrogen. Now, why would we use nitrogen? What's DNA made of? Nitrogenous bases. So if we use nitrogen 15, all the nitrogen in the new strands made from then on are made of what? Nitrogen 15. So they're heavier than nitrogen 14. Does that make sense? Remember, these are isotopes, which means this has one extra neutron, right? All right. Then we could take those cells. By the way, these little, they're not cells, they're colonies. There's millions of bacteria in this colony, this dot. There's millions of bacteria in this dot, and many dots here. We call these things plaques, which is why when you go to the dentist and they remove the plaque, they're removing colonies of bacteria growing on your teeth. Doesn't it gross you out? Grosses me out.
I like that.
You do? So we can take those bacteria, we can put them in a test tube. We break them apart like we did with the heat kill. Break them apart, and then we put them in a centrifuge. And it turns out the nitrogen 15 is going to be radioactive, so we can find out where it is, right? So we can put in a band, and when we put it in a centrifuge, remember it's like a spin cycle of a washing machine, remember that? And the heaviest stuff will sink to the bottom, the next heaviest stuff will be in the middle, the lightest stuff will be at the top. Both test tubes. Agreed? If we did that and we spun them in a centrifuge, what you'd find is the DNA with the nitrogen 14 would be up here. The DNA with nitrogen 15 would be down here.
Because like it's more.
It's what? It's denser. The more dense the material, the lower down in the test tube it is. Because all the nitrogen in the DNA is made of nitrogen 14. It's less dense. It's up top. All the nitrogen all the DNA is made of nitrogen 15. It's more dense. It's lower down in the test tube. That's important that you understand. If you don't understand that, you're not going to understand the experiment. And people were like, "This is hard to understand." I said, "Yeah, you got to take it one step at a time. See how complicated this is, and yet simple." Look and see, what are we going to do with the experiment? What we're going to do is we're going to allow them to have babies. We're going to take those colonies, this was grown in nitrogen 14, right? Let's say nitrogen 15. Let's do nitrogen 15 first. And we're going to put them in a test tube that only has nitrogen 15 food. So they're going to put that in. So the original DNA has what? 2 strands, right? They're both made of nitrogen 15. All the nitrogen is nitrogen 15. Everybody agree with that? Okay with that? That means 7 protons and 8 neutrons, right? Because it's an isotope. And in the test tube, I put nitrogen 15, that's my bad. In the test tube, you're only going to have nitrogen 14. So if it's conservative, let's look at the 3 possibilities it could be. If it's conservative, which means that the children, that 2 of the cells right? There's gonna be 2 cells every time it divides. 1 cell will have what? Conservative means 1 cell will have nitrogen 15 each, right? There'll be copies, but one only has the original is nitrogen 15, and the other one's gonna, because they're conserved, the new one's gonna be what? Made of what? Nitrogen 14. So we expect to see how many, if it's conservative, right? We would expect to see what? How many bands? 2. You got it right. 2 bands. 1 nitrogen 14 band, right? After we break up the cells and run them in through a centrifuge and separate them, and 1 heavier band, the nitrogen 15 band. That's what we would expect to see if it's conservative.
So all
Yeah.
spinning in that little tubey thing you were talking about?
Yeah, the spinning in the tubing thing. That's right. So we spin them in these test tubes in the centrifuge, and we'd expect to see first generation, the first time they multiply, the first event, the first reproduction event, first time the cells make copies. We'd expect to see 2 bands. If it was dispersive, then we'd expect to see what? In other words, some of the nitrogen 14 is going to be mixed with the nitrogen 15 about evenly. We'd see how many bands? Think about it. You have nitrogen 15, the 2 new cells. Let me draw it out for you. Then if it was, so this is what would happen if it was conservative. If it was dispersive, what we would expect to see is the nitrogen 15 here, 2 nitrogen 15s to begin with, and the 2 new ones would be some nitrogen 15 and some what? It's nitrogen 14. So there'd be kind of a mix, right? And we'd expect to see that in both of the new cells. Does that make sense? So how many bands would you expect to see if they happened about evenly? How many bands in the dispersive model? 2 of each. So you started with 100% nitrogen 15, and now you have about 50% nitrogen 15, and here 50% nitrogen 15. So how many bands? Wouldn't they be the same mass? So how many bands? When you centrifuge it down, it'd only be 1 band. You'd expect to see only 1 band. Instead of seeing 1 band in nitrogen 14 here, and 1 band in nitrogen 15, because in the conservative model you'd have one that's 100% 15, the other 100% 14. Here you have 50-50 each, so they're both the same mass, so you only have 1 band. So what you're missing is this stuff, right? You're missing the higher, it's in the middle, right? You're missing the bottom and the top. It's not 14 or 15, it's somewhere in between, right? It's not 14 or 15, it's somewhere in between. If it were dispersive, right? Somewhere in between. Do you agree? There'd be 1 band somewhere in between. Whatever that is. Now, if it was semi-conservative, meaning it's you have 15 here, right? Nitrogen 15. And you have 2 babies, 2 new cells. And what would you have? One that would be 15, and what would the new strand be? The other would be 14. And so, but that would be true for both the new cells, right? That would be true for both the new cells, nitrogen 14 and nitrogen 15. Does everybody agree with that? So how many bands would you expect to see in this one when you centrifuge it down? Don't put anything away. Some people put stuff away already. They're not looking at the board. They're not writing anything down. My guess is they're not going home. They're not going to go home and watch the video either. You're dismissed when I dismiss you. All right?
Wait, did you mean to write?
How many? Yeah, so this is 15 and 14, 15 and 14. Now, where the dispersive, each strand is all broken up, about 50-50. In the semi-conservative, you have 1 strand is old and 1 whole strand is new. So this is semi. How many bands do you expect to see in the test tube?
2.
Why 2? Wouldn't you expect to see 1 right between 14 and 15, halfway? Because half is 15, the other half is 14 for each of these. Aren't they the same mass?
Right.
Right? So we only have 1 band, and you have 1 light one, 1 heavy one. It's not the light ones, the heavy ones aren't there. Just like what? Just like the dispersive model, right? So there should be no difference between the dispersive model and the semi model in the first generation. Wait, we're not going to do it. You're going to finish this. We're going to finish this. But look at this. Let's look at this. What we found in the experiment is that the results, now this is what we predicted, the results were this. 1 band halfway between the 14 and the 15. So which one is it not? It's not conservative. So it's not this one, for sure. But we don't know which one of these other 2 it is. So we let it run again, with more nitrogen 14 again. Now, if you're going to have it run again, you're going to make 2 new babies, right? Yeah, you're going to do it again. You're going to take that, not with the conservative. The conservative's already been ruled out, right? So now we have the dispersive model. What's going to happen if we have the dispersive model? What are you going to make? You're going to make, again, half and half, right? With equal mass. Do you agree? It's always going to be half and half with equal mass. So you're always, with a dispersive model, you're always going to have, what? 1 band in between the heavy and the light. Because you're always going to have some of that heavy in there. It's not ever going to be as light as a pure nitrogen 14. Does that make sense? All right. So what's going to happen with the semi-conservative model? No, it won't be. Think about it. That's why I want you to stay and think about this. So now if you take this, which is half and half, and you're growing it in nitrogen 14, remember the light one's nitrogen 14, the black one is nitrogen 15. So nitrogen 14, nitrogen 15, you make copies, you only have nitrogen 15, you're going to keep one of the originals, right? So, but one of the originals is what? Nitrogen what? 14. The other one is nitrogen 15, so that's good. I'm sorry, that's wrong. One of the originals is nitrogen 14, that's great. But then, of course, the new one's also nitrogen 14, right? Does that make sense? And then the other one's going to have one of the original strands, but it's going to have a new one that's nitrogen 14. So what bands do you expect in the second generation? You expect to see 2 bands. Excellent. One is going to be where? Is there any going to be a nitrogen 15?
No.
None are going to be a nitrogen 15. So there's none at nitrogen 15, because there's nothing that's pure nitrogen 15 here. We're going to expect to see 1 band up here at where? At nitrogen 14, and 1 in the middle. That's in between, that's 50% nitrogen 14 and 50% nitrogen 15. Does that make sense? And in fact, guys, this is the answer. This is what they found. The first generation was in the middle. They couldn't tell the difference. They did it again, and they found 2 bands, one in nitrogen 14, one in nitrogen 15, which means that the way we replicate DNA is called semi-conservative. That's the experiment. That's explained the whole thing. And it's all drawn for you in the project. Please answer the questions and come tomorrow with some questions in class. Have a great day. Oh wait, announcement. Announcement. Anyone in I Am Bella, please meet in the dance room. After you check in, I'm assuming they mean after you check in attendance. All right? If you don't know, you're not part of the group. All right, have a good one. Beautiful at the same time. Amazing.
Oh, back. Hold up.
So, as I keep telling you repeatedly, This is being recorded. I will post it, and when you post it, if you need more time, you can go back and look. It is 3:15. I literally have a couple minutes to finish this. So nitrogen 15, if I took this bacteria with all the DNA is nitrogen 15, I let it do its duplication, but I only give it what? Nitrogen 14, so all the new strands will be nitrogen 14. All the old strands will be what? Nitrogen 15. Okay, so let's go ahead and do that. If it's conservative, all the old strands would stick together, so they would all have 100% nitrogen 15, right? And all the new strands would be nitrogen 14. After one round of replication, how many bands would I expect to see in the test tube after I spun them down? Individual bands in the test tube. Why 4? No. I'd expect 2 bands. Why would I expect to see 2 bands? Because half the DNA has nitrogen 15 because it's conservative. The other half has nitrogen 14, so the nitrogen 14 is going to be higher up in the test tube than the nitrogen 15. That's what I would expect to see. It's 2 bands. Do you understand that? After I spun them down. Now what if I had the dispersive model? In other words, I had all of it nitrogen 15 at first, then after I grew them, parts of each strand were nitrogen 14, parts were nitrogen 15. So 50% of the DNA would be mixed, but they would both be pretty much the same density. How many bands would I expect to see if they all have the same density? 1 band, that's right. I'd expect to see only 1 band. Why would I expect to see only 1 band? Because 50%, both of them, they'd be 50-50. All the DNA would be 50% nitrogen 14, 50% nitrogen 15. So I'd expect to see 1 band. In between 14 and 15, they wouldn't be as high as 14 or as low as 15. Do you agree? Yeah. All right. The last one is a semi-conservative model. If we had a semi-conservative model, you had 100% nitrogen 15. When you're done, half the DNA would be 15. The other half would be 14. For both samples, both of the new cells would have that. So after I broke it down and spun it, I'd only expect to see 1 band again, right? Right? Because it's still 50-50. Well, the result was this. The result of the experiment was it was 1 band. So what did that tell you? Hold on, we're not going anywhere until this is done. So we have there's that 1 band. So we know it's either dispersive or it's what? But we know it wasn't what? It was not conservative. Now the next part of the experiment, please wait. You did the same thing over again in the second round. And if you think about it, in the second round, if it's dispersive, then you got 50-50 in the original. You're going to have 50-50 in each of the progeny. And so when you dissolve it and when you dissolve the cells and you spin them, you'd expect to see 1 band again, wouldn't you? In the semi-conservative, this is the trick. Listen, because you got to go back and look at this. It's going to be on the test. It's the last thing I got to say, then you can leave. Just listen to me. If you have this half nitrogen 15, half nitrogen 14, and you do DNA replication, half of the new cell, the DNA will be all nitrogen 14, because look, in the second round, one of the strands is what? And then the new strand is still what? One of the half the DNA is nitrogen 15, and the new strand is what? So you got half this. I hate the fact that people are putting stuff away, and you're in a hurry, but it would go a lot faster if I wouldn't have to stop to tell you don't put stuff away. We'd be gone already if I didn't have to stop. So this is both nitrogen 14. This is half nitrogen 14, half nitrogen 15. How many bands would I expect to see if it's semi-conservative? 2. One at nitrogen 14 level, and the other one in between 14 and 15, right? So what we saw in the second round of replication was this. So the way we replicate DNA is the semi-conservative model. We all understand that? This will all be tested, finish all those questions on that project, have a great day.